from pixels to proteins. My journey from ultrasound to immunotherapy. By Rob Entrekin. I want to tell you a rather unlikely story about how an old guy like me ended up doing cancer research in a cutting-edge medical institute at age 75. This is a word we all know far too well. If you haven't been diagnosed yourself, you have a parent, a spouse, a sibling, a friend, a child, or a colleague who has. Cancer is the second leading cause of death in the United States. About 1 in 3 of us will be diagnosed with it in our lifetime. About 2.1 million Americans will be diagnosed with cancer in 2026. That's almost 6,000 new diagnoses every single day. Over 600,000 of us will die, about 1,700 every day. But here's the good news. The 5-year survival across all cancers has gone up from 49% in the mid-1970s to 70% today. That's because of decades of research. And that's why research matters. It changes the survival curve. The number of children diagnosed with cancer in America is much smaller, about 15,000 a year. But pediatric cancers are biologically different. The genes involved are different. A treatment that works on a 60-year-old often does not work on a 6-year-old. Because pediatric cancer is rare compared to adult cancer, Big Pharma typically can't justify the cost of developing drugs specifically for kids, which means somebody else needs to do it. This is where I work. Building Cure is one of 3 buildings in downtown Seattle that comprise Seattle Children's Research Institute, the research arm of Seattle Children's Hospital. SCRI has about 1,700 employees, including 340 senior scientists known as principal investigators. They're all doing research dedicated to finding new treatments for childhood diseases. My lab is part of the Bentown Center for Childhood Cancer Research. The Bentown Center exists because a Seattle family lost their son to neuroblastoma in 2008. His parents had the financial means to do something about it. They funded a research center within SCRI focused entirely on finding cures for childhood cancers. The cancers Big Pharma was not investing in. This is my boss, Dr. Jim Olson, MD, PhD. As an MD, Jim is a pediatric neuro-oncologist, which means he treats children with brain cancer at Seattle Children's Hospital at the Sandpoint campus. And as a PhD, he's also a scientist and a principal investigator. He runs a research lab of about 25 scientists and technicians who design and test new drugs to treat pediatric brain cancer. You'll hear more about Jim shortly. The obvious question is, what the heck is a 75-year-old retired guy doing in a pediatric cancer research lab? I'm not a physician. I don't have a PhD in chemistry or pharmacology. I've never even taken a course in biochemistry, immunology, genetics, or molecular biology. So why am I working there instead of fishing or playing golf in retirement? That's the story I'm going to share with you now. Let's start at the beginning. Ever since I was a kid, I loved science. I was a teenager during the space race of the 1960s, and I turned 18 just 4 days after Apollo 11 landed on the moon. In college, I trained as a biomedical engineer. I earned my bachelor's degree at Rose-Hulman and 2 master's degrees, one at North Carolina State and one at Duke. It was 50 years ago at Duke that I first learned about the emerging field of diagnostic medical ultrasound imaging, and I was fascinated by the technology. That was the beginning of my career in ultrasound. After Duke, in the autumn of 1978, I joined a small startup company in Bellevue, Washington, called Advanced Technology Laboratories, or ATL for short. ATL's ultrasound technology was a spin-off from the University of Washington, and the company was growing exponentially. from about 50 employees in 1977 to over 1,000 employees 5 years later. I was employee number 100. By sheer luck, ATL turned out to be an ideal place for a young biomedical engineer. I was in the right place at the right time to grow my career as part of a rapidly expanding company in the burgeoning field of medical ultrasound imaging. I loved my job. For the next 2 decades, ATL was wildly successful, growing their annual revenue from about $5 million in 1978 to over $500 million in 1998, a 100-fold increase, about 26% growth every year for 20 years in a row. In 1998, ATL was acquired by Philips, the Dutch electronics giant. I continued in my role as a technical fellow, performing research and development of new ultrasound imaging technology for an additional 24 years. I was just a month short of my 71st birthday when I retired from Philips. You can see from the photos there's been a bit of wear and tear, and a lot less hair over the past 50 years. Ultrasound imaging has made tremendous progress over the past 50 years, driven by the revolution in microelectronics and computer technology. What began as fuzzy blobs flickering across an analog cathode ray tube evolved into millions of crisp digital pixels, rendered in real time on a computer screen. When I started in 1975, ultrasound scanners could barely produce a grainy black and white image of a fetus's head, just enough to measure the size of the skull to confirm the baby was growing on schedule. 25 years later, those images had hundreds of shades of gray and 10 times better resolution. A doctor could not only count fingers and toes, they could perform a complete physical examination before birth, checking that the baby's heart, spine, brain, liver, kidneys, and lungs were all developing normally. 50 years later, parents take home a 3D video of their baby before they're even born. During my career, I've invented many techniques to enhance ultrasound image quality and automate measurements, giving doctors more accurate and useful tools for diagnosing disease. For example, I helped design ultrasound systems that painlessly detected breast cancer that X-ray mammography missed. With my colleagues at Philips, we developed image processing software that measured aortic aneurysms more precisely than doctors can. In my final few years, I led a team that developed artificial intelligence software that automatically finds and measures atherosclerotic plaque in the carotid arteries, which is an early warning sign for heart attack and stroke. Along the way, I had the privilege of working with world-class radiologists, cardiologists, and vascular surgeons in the United States, Denmark, Spain, France, Germany, and the Netherlands. Together, we designed and ran numerous clinical trials to validate these new technologies, many of which were subsequently approved by the FDA and launched as products. In 2007 and 2008, while I was still working full-time in ultrasound R&D at Philips, I earned a certificate in winemaking through a 2-year online program at Washington State University. In the fall of 2008, I launched a small, commercially licensed and bonded winery from my garage in Kirkland called Finn Hill Winery. For 14 years, I was running 2 careers in parallel: engineering by day, winemaking by night. And it was the winery that led me into the world of charity fundraisers. In 2013, a neighbor invited me to donate some wine to help raise money for Camp Good Times. Camp Good Times is a summer camp on Vashon Island for kids with cancer or who are cancer survivors. It's a camp where children who are actively receiving treatment for cancer can have fun and just be regular kids for a week. It's operated mostly by volunteers, and it's been serving the pediatric cancer community for over 40 years. So, of course, I started donating Finn Hill wine for their fundraising events. I even made special large format bottles with the Camp Good Times logo etched and painted on them for their silent auctions. This is how I got connected to pediatric cancer in the first place, because I wanted to support the kids. In 2015, I went to the Camp Good Times annual gala fundraising dinner. The keynote speaker that night was a pediatric oncologist from Seattle Children's, Dr. Jim Olson. He gave a talk about his passion for trying to find cures for childhood cancers. He talked about Project Violet, a community crowdfunding initiative to raise money to support their research at Fred Hutchinson Cancer Research Center. Before we go on, I want you to hear directly from Jim. The video I'm about to play isn't from that 2015 gala. It's an excerpt from a TED Talk Jim gave in 2013, but it's the same story about the same mission. I want to give you a sense of what I experienced that night in 2015.
When I was a medical student, I was walking home through the Arboretum in Ann Arbor one day, and I had a really light step, and I was actually kind of singing to myself. And those of you who know me wouldn't be too surprised by that because I'm generally an upbeat person. But that otherwise could have been the worst day of my life so far. You see, that day, it was the first time ever that I was caring for a little child who died under my care. She was a beautiful little 7-year-old girl who each day lost more neurologic function until finally she was unable to breathe. And her family decided not to prolong her life, but to let her go gracefully and lovingly. And at the end of that afternoon, before I left the hospital, her parents sought me out. And they said they had something they wanted to share with me. They said that I taught them that her first breath was an utter miracle, and that every breath after that was icing on the cake. That I taught them that it didn't matter if her life was 7 days or 7 years or 70 years. What mattered what was between the beginning and the end. And they wanted me to know that even when medicine didn't go the way we wanted it to go, that I had a gift to share with families of children. And you can imagine what that feels like when you come upon an x-ray like this going into clinic. And you see that in this beautiful brain of a child, there's a huge tumor. That big white thing in the middle isn't supposed to be there. And that's the brainstem that connects the brain to the rest of the body. In my 23 years of practicing oncology, I haven't yet ever had a child survive with this cancer. And so I have one hope. And my one hope is that before I retire, that I can walk into a room of a child that has an x-ray like the one I showed you and offer them real hope. That we have a therapeutic that could save their life and allow them to grow to be an adult. Now, Violet also had a brainstem glioma, and she knew she was going to die. She was not afraid of death, and she and her family asked if, at the time of her death, we could take her to autopsy and to remove her brain tumor and to create research tools from it and share them with scientists around the world so that other kids would not have to go through this in the future.
As you can imagine, I was deeply moved by the message and the mission that Jim and his research team were pursuing. After hearing him speak that evening, I introduced myself to Jim and asked, "What can I do to help?" So of course I started donating Finn Hill wine to Project Violet to help raise money to support the Olson Labs research, alongside the wine I was already donating to Camp Good Times. This was really the same cause from two different angles. Camp Good Times to support the kids, Project Violet to support the research. So for the next 5 years, I worked full-time at Philips in ultrasound engineering, and on evenings, weekends, and vacations, I made wine at Finn Hill Winery, and I donated special bottles of wine to both Camp Good Times and Project Violet. Pediatric cancer was now an important part of my life. I've told you about my career in ultrasound, my second career as a winemaker, my connection to Camp Good Times and Project Violet, and how I met Jim Olson. What I haven't told you yet is how I actually ended up working in his lab. That took a few more years and 4 major events. In 2020, COVID-19 brought the world to a standstill. Charity galas were canceled. Camp Good Times paused. Project Violet events stopped. The Finn Hill Winery tasting room was closed indefinitely. I continued working at Phillips remotely through video conferencing. I knew my retirement was on the horizon, and nobody had any idea how long the COVID lockdown would last. Given that uncertainty, I decided I wasn't going to make any more wine in 2020, so 2019 ended up being Finn Hill's last vintage. For the next several years, I just focused on selling through the bottled wine inventory I already had in the warehouse. That whole parallel life I had built, the winemaking, the charity work, the connection to pediatric cancer, all of it just went quiet. In 2022, my world changed again. I finally retired from Phillips in June, just a month shy of my 71st birthday. So now my ultrasound career and my winemaking career were both over. Over the following year, I settled into a new routine, going to the gym 3 times a week, taking cooking classes at PCC, and growing dahlias and roses in the garden. Then midway through my second year of retirement, I got bored. I didn't want to go back to work as a consultant in medical ultrasound imaging. I'd been doing ultrasound for over 40 years, but I missed being involved in science and technology and healthcare. I wanted to find a worthy cause where I'd be helping people and serving the greater good, and most important to me, learning something new. So in the spring of 2024, I reached out to Jim, who had moved his research lab from Fred Hutch to Seattle Children's. We met for coffee, and I asked him the same question I'd asked him in 2015 at that Camp Good Times gala: how can I help? And he said, we have far more work in the lab than we have hands to do it. If you want to come do bench science, we can train you. I jumped at the chance. So in June of 2024, I was hired as a summer intern, just a month before my 73rd birthday. Over 2 years later, I am still classified as an intern. The HR code on my paperwork is literally the one they use for visiting college students. Nobody's bothered to change it. I get paid minimum wage, but honestly, I would do it for free. So that's the story of how I became an intern in a cancer research lab at Seattle Children's. Now let me take a few minutes to give you a brief glimpse into the science behind a new immunotherapy drug. Immunotherapy is a new type of treatment that uses your body's own immune system to target and destroy disease, such as cancer. It adds a fourth option to the traditional cancer treatments of surgery, chemotherapy, and radiation. 2 years ago, everything I'm about to tell you was a complete mystery to me, and I'm still learning. Your immune system is not an organ like your heart or your liver. It's a complex system, possibly the most complex system in the human body. It consists of trillions of cells scattered through your blood, your lymph nodes, your gut, and your skin, all communicating with each other. Its job is simple to state, but almost impossibly complicated to do. It must distinguish you from not you and destroy anything that's not you, for example, bacteria, viruses, or even cancer cells. To understand the immune system, we need to start with proteins. You certainly know about proteins from food nutrition labels. But proteins are important for a much bigger reason than the ingredients on a box of cereal. Proteins are the body's workforce. About 17% of your body weight is protein, but the more important fact is that almost everything that happens inside you is done by a protein. Proteins digest your food, they carry oxygen in your blood, fight infections, contract your muscles, heal your wounds, read your DNA, and send signals that let one cell talk to another. If something is getting done in your body, a protein is almost certainly doing it. And every protein is built from a set of 20 building blocks called amino acids. Just as letters of the alphabet spell out a word, amino acids link together to spell out a protein. Proteins range in size from a couple of dozen amino acids to over 30,000. They snap together like blocks in a long chain called a polypeptide. But these blocks are not inert. Each amino acid carries its own electrical charges and chemical properties, so different parts of the polypeptide chain are attracted to or repelled by one another. Once the chain is assembled, those forces cause it to fold into a complex three-dimensional shape. That folded chain is the finished protein, and the folded shape of that chain is everything. If you rearrange the sequence of amino acids, or change the length of the chain, or substitute even one or two amino acids, you change the three-dimensional folded shape and therefore the function of the protein. That's how the body builds thousands of different proteins out of the same 20 building blocks. Form equals function is a fundamental principle of molecular biology. Every protein does what it does because of the three-dimensional shape it folds into. Get the shape right, and the protein works. Get it wrong, and it doesn't. Your immune system uses that same principle to check a cell's ID. Here's a normal, healthy cell. Inside, it's always building proteins. It chops up those proteins into short peptide chains called antigens. The cell continuously pushes those antigens to the surface on platforms called MHC. A single cell can post thousands of antigens like this, all out for inspection, where they act like molecular barcodes. Your immune system has a squad of inspectors called T cells that patrol your body around the clock, checking every cell they meet, reading those surface antigen barcodes to identify each cell as friend or foe. The T cell reaches out with a special receptor that works like a barcode scanner. It reads the antigen to tell whether this cell is part of you or something that isn't. This cell reads as normal, so the T cell lets go and moves on. Here, in blue, are real T cells under the microscope, sped up 100 times. What you see in this 15-second video actually took almost half an hour in real time. The clear cells are all normal and healthy, so the T cells scan each cell's barcode, then move on. This is happening inside you right now all the time. It's just routine maintenance. So we've established that the immune system reads the cell's barcodes. Now let's watch what happens when it finds a cancer cell. Cancer begins with a mistake in a cell's DNA called a mutation. And since every protein is built from the instructions in a cell's DNA, mutated DNA makes abnormal proteins. The cancer cell displays corrupted antigen barcodes that no healthy cell would ever show. The T cell scanner locks onto the cell and reads the barcode. The displayed antigen is corrupted and marks it as a cancer cell. When it reads the corrupted barcode, the T cell transforms from an inspector into a killer. The killer T cell releases a burst of toxins that punch holes in the cancer cell, triggering its destruction. And here's the remarkable part. A killer T cell doesn't stop at one. It kills, then moves on, finds the next cell with that corrupted barcode, and kills again. It's a serial killer, and that's exactly what you want it to do. This next clip is the real thing under a microscope. The T cells are the green ones. Watch a single T cell attack one cancer cell, then move on to the next and the next. Each cancer cell it touches turns red as it dies. Your immune system does this constantly, finding damaged and abnormal cells and clearing them out before they ever become a problem. Which raises an obvious question. If your body can spot a cancer cell and kill it like this, why does anyone ever get cancer? We've got T cells that can destroy cancer cells, so you'd think cancer wouldn't stand a chance. Why do we still get it? With trillions of cells being inspected around the clock, the immune system needs a built-in way to hold its fire, so it doesn't attack its own healthy tissue by mistake. That safeguard is a real protein called PDL1. Healthy cells hold it up like a flag that signals, "Don't shoot, I'm part of you." A matching receptor on the T cell called PD1 sees that flag, and seeing it puts the T cell on safety, like the safety switch on a gun. The gun is still loaded, but it can't fire. So here's the problem. Cancer cells start out as part of you. They're your own cells, gone wrong through genetic mutation. And many of them retain the PDL1 flag. When a T cell inspects one of those cancer cells, it picks up two signals. It reads the corrupted barcode, exactly as we saw before, which would normally be the order to fire. But it also sees the PDL1 "Don't shoot me" flag, so the T cell goes on safety instead. It stands down and moves on. Now the question is, how do you get a T cell to attack a cancer cell that's waving a "Don't shoot me" flag? This is where protein engineering comes in. Our lab develops proteins called T cell engagers. These molecules have a clever way to bypass the PDL1 safeguard. They can take many forms, but the simplest version is called a bispecific T cell engager, or BiTE for short. This is the first BiTE ever approved for patients. It's called Blincyto, made by the pharmaceutical company Amgen. What you're looking at is a model of its molecular structure. Schematically, it's beautifully simple, just two binding ends connected by a flexible linker. One end is shaped to lock onto a particular antigen on the cancer cell. Ideally, that antigen is far more abundant on cancer cells than on healthy ones. The other end locks onto a receptor on a passing T cell. In this case, Blincyto grabs the cancer cell by an antigen called CD19. Then it catches a passing T cell by its CD3 receptor. CD3 is the T cell's trigger, the part that fires it into action. The BiTE sticks the two cells together face to face. The T cell switches on and destroys the cancer cell with its toxins. The T cell also multiplies and calls in reinforcements, turning a few engaged T cells into an army of killers. When the T cell is freed up, it's still armed with the BiTE and ready to go find its next target. And notice what the BiTE does here. Normally, the T cell only pulls the CD3 trigger after it reads an abnormal barcode, but if it also sees the PDL1 "Don't shoot me" flag, it goes on safety and doesn't fire. But the BiTE skips all of that. It grabs the CD3 trigger and pulls it directly. When both ends of the BiTE have locked on, the trigger is pulled, and the attack happens no matter what the cancer cell is displaying. Blincyto is the simplest kind of T cell engager, with just two ends. But the same idea can be built into far more sophisticated molecules, engagers with 3, 4, or even more binding ends. And that's exactly the kind of thing my lab designs and tests. When I started my career, immunotherapy didn't exist. T cell engagers barely existed 10 years ago. Blincyto was the first, back in 2014. But in just the past few years, about 10 more have won approval, and dozens more are in clinical trials. Today, they mostly treat cancers of the blood, lymph nodes, and bone marrow. But current research is developing new molecules that can go after solid tumors in the lung, prostate, breast, and liver. The Olsen lab basically does three things. It designs, produces, and tests new cancer immunotherapy drugs. These drugs are all various types of T cell engager proteins. Based on the specific type of cancer, our scientists design new T cell engager molecules to help the immune system find and destroy the tumor. They use AI software tools to design the molecule, so it folds into the correct 3D shape. When the design is complete, the amino acid recipe for the engager is then programmed into living cells grown in the lab, which act as tiny factories to manufacture the protein. We separate the target protein out of the cellular soup and purify it, sorting the molecules by what they stick to and by their size. Finally, we perform preclinical testing of the new drug to find out if it kills cancer cells in a dish, and if so, at what dose. If it works well in a dish, we then study whether the drug kills cancer in mice and whether it has any harmful side effects. But even if a drug works well in mice, we can't be certain that it will work well in humans. So the most promising drug candidates are licensed to biotechnology companies for human clinical trials. So where do I fit into all of this? In the 3 stages we just discussed, I work in that middle stage where we produce, purify, and verify the proteins. First is production. Remember those living cells that act as tiny factories? I help grow them, feeding and expanding the cultures until they've made enough of our therapeutic protein, which typically takes a couple of weeks. Then we harvest the liquid growth medium that contains our target protein along with thousands of others. As an analogy, think of the liquid growth medium like a gallon of vegetable soup, and all we are interested in is the carrots. Next comes purification. That's me in front of a machine called an ACTA liquid chromatography system. It works like a very sophisticated filter. The vegetable soup flows into the top of that glass column, which is packed with microscopic beads. Our target protein sticks to a special coating on the beads, and everything else washes through the column and down the drain. Then we add a buffer solution to release the target protein from the beads, and collect about a tablespoon of liquid containing just a few milligrams of the protein. It's like the whole gallon of soup only had a tiny sliver of carrot to begin with, and that tiny sliver is the only thing that we filter out of the soup and keep. Last, quality control. Is the target protein the right size? Is it pure? Is it safe? One of my main tools, gel electrophoresis, is a kind of protein fingerprint that sorts the sample into bands by the size of the molecules. When the right bands show up in the right places, I know we've made the right protein. And finally, I perform a safety test to ensure the protein is free of bacterial contamination before we start testing it in living cells or animals. My job is just a small part of a much larger effort, but every batch of protein I prepare is one step closer to a medicine that might help a child. I'm 75 years old, and officially I'm still classified as an intern. In my career as a biomedical engineer, I felt deep satisfaction knowing that my technical contributions helped doctors diagnose and care for millions of people around the world. In my career as a winemaker, I took pride in crafting a good bottle of wine that brought a little cheer into people's lives. But my job at Seattle Children's is different. I'm inspired by the mission, and I'm grateful to play even a small part in helping these brilliant scientists develop cures for children facing life-threatening illnesses. And for me, the icing on the cake is getting to learn something new every day. After 44 years of being the ultrasound expert in the room, I'm a student again, and that has turned out to be a gift. Of all the things I could be doing with this chapter of my life, this is the one that called to me, and I'd choose it again because it's another chance to make a difference in the world. I don't have any plans to retire again, because being an intern is one of the best jobs I've ever had. If anything I've said has inspired you, here are 2 ways you can support the ongoing efforts to help kids with childhood cancers. Beyond money, these organizations gladly welcome in-kind donations or donations of your volunteer hours. Thank you for listening.